Hi everyone, good afternoon for those that are in Brazil. We are unfortunately come close to the end of our symposium, but we still have very nice talks. And now we have, I am very happy to introduce you for Dr. James Bounce. Dr. Bounce is a researcher on USDA for more than 40 years. He has several works on the area of photosynthesis and stomatal conductance, and now he is a consulting on PP systems. So he will be talking here a little bit more on the PP systems. And I would like to say thank you, Dr. Bunce, to accept our invitation, and also for PP Systems for this sponsorship and to this possibility, and also to Lab Control to intermediate this contact and make possible this talk today. So, Dr. Bunce, please feel free to start as we wish.
All right, thank you. First of all, I'd like to thank the organizers of the Brazilian Symposium on Photosynthesis for inviting me. As it was said, I retired from USDA about 4 years ago, and I've been continuing some work at USDA and also doing consulting work for PP Systems, sort of doing technical and applications support for them, and also working with their development and engineering team. The work I'm going to talk about is actually work that was conducted at USDA and using PP Systems instruments, but other than that, I won't say very much about PP Systems in this talk. So what I'd like to talk about is photosynthetic responses to brief high and low temperature events in crops grown at ambient and elevated CO2. And you might think it's odd that I'll talk about low temperature events. And the reason for doing that is probably not obvious. And that is, the reason is that farmers in temperate climates who are trying to avoid the hot spells in the middle of the summer are trying to plant crops earlier and earlier in the spring, where they run into low temperature problems. So, and there has been some literature suggesting that elevated CO2 can protect plants from low temperature damage to photosynthesis. And I'll talk about that partway or in the second half of the talk. Back in 1991, Steve Long pointed out the fact that C3 photosynthesis models predict that elevated CO2 raises the optimum temperature for photosynthesis. And this is just a plot of assimilation rate versus leaf temperature at 3 CO2 levels. This is not a particularly good figure, but it's directly from his publication. And you can see that at 350 parts per million, which was ambient back then, the optimum temperature was a little over 20, 22 or so. And then as you went up in CO2 concentration, up to 650 was the high one here, that the optimum temperature actually increases. So these are just predicted light saturated rates of CO2 uptake. And so when photosynthesis is limited by Rubisco carboxylation capacity, then stimulation by elevated CO2 increases with temperature, as laid out in the Farquhar, Van Kemmer, and Berry model. And this is just a graph for showing assimilation rate versus temperature at a CI, internal CO2 concentration of 250 or 350, so sort of ambient and elevated CO2, when photosynthesis is limited by the maximum carboxylation capacity, VC max. So you can see that these curves, as we go up in measurement temperature, the curves diverge. So obviously, there's a larger stimulation in photosynthesis at higher temperatures than at cooler temperatures. And this is what Long was talking about. There is a problem with this simple analysis, and that is that if electron transport actually limits photosynthesis at elevated CO2, the stimulation may disappear as temperature increases. So this has the same two initial figures of at CI of 250 or 350 ambient and elevated CO2, limitations by VC max. But if you're at 350 parts per million for a CI and J becomes limiting, then this red line shows that as you go up in temperature from that, the assimilation rate actually goes down. So whether high CO2 increases the optimum temperature for photosynthesis is going to depend on whether photosynthesis at elevated CO2 is limited by VC max or whether it's limited by J. So for global warming and rising CO2, so a big question is what actually limits photosynthesis at elevated CO2, either at the current or at warmer temperatures? Is it VC max or is it J? And unfortunately, there's very limited information about that, especially for warm temperature treatments or especially also in the field. So there's as I will do throughout here, what I'm doing is pointing people in a direction of places where there's obvious a need for more research rather than giving you answers about what research says. So to figure out this question of what is limiting photosynthesis at high temperatures, is it at elevated CO2? Is it VC max or is it J? Obviously, the solution to that, for the way to answer that question, is to do ACI curves under the growth conditions. And I would point out that now these can be done much faster than they used to be able to be. Used to be a very slow and tedious process, but now we can do ACI curves, complete ACI curves in 5 minutes with a PP system. So I'm going to switch now from to talk more about brief high temperature stresses. And what I mean is temperatures at which photosynthesis becomes, if you hold it at that temperature, photosynthesis just gradually decreases over time. So it's a stress enough to substantially limit photosynthesis and cause some more than temporary injury to photosynthesis. There are three things that may be going on when you're talking about brief high temperature stresses. It could be that rubisco becomes deactivated by the high temperature. And then the question is, does elevated CO2 have an effect on that deactivation? And if we're talking about deactivation of rubisco by high temperature, then that should be a reasonably quickly reversible issue. So when you return temperatures back to normal, the activation state should come back to normal. So the question is, to what extent is that happening? Another possible thing that can happen is that there's a disruption to electron transport systems. And again, the question is then, would there be a CO2 effect on that? And would that be more of an irreversible damage than just a deactivation of rubisco? And the third thing is membrane leakage. Is there a CO2 effect on that? And again, how reversible is that kind of damage to brief high temperature stresses? So this is just some literature. This is a publication by Wang et al. And this is just different species grown and kept at either 700 parts per million or 370 parts per million. And looking at during a duration of a heat stress, whether photosynthesis is inhibited more or less at high CO2. And you can see in a case like this, where the difference in photosynthesis initially with an elevated CO2 having higher photosynthesis, both of them come down, but the difference between them disappears. So there's actually more inhibition at elevated CO2 than there was at ambient CO2. And that's certainly the case here in the C4s, where, as expected, they're not very different under normal conditions, but the heat stress actually inhibits photosynthesis more at the higher CO2 than it does at the ambient CO2. And here's another case where the inhibition is actually larger at elevated CO2 than it was at ambient CO2. So certainly there's not always the case that high CO2 is going to protect photosynthesis. Some other literature, Taub found that elevated CO2 reduces the decrease in chlorophyll fluorescence that happens at high temperature in several C3 species. So there are no real photosynthetic measurements here. It's just chlorophyll fluorescence issues. Maize et al. Found for 2 tree species that elevated CO2 in open-top chambers actually reduced the damage to photosynthesis caused by high-temperature stress. And then another paper with Maize by Co2. And actually here, elevated CO actually increased the damage caused by high-temperature stress. These and the early literature seldom have tried to separate the effects of growth CO2 from immediate CO2 effects. And the experiments I'm going to present to you have attempted to do that. So we have plants that are grown indoors in controlled environment chambers at this temperature, 29 day and 17 night, at high light, and CO2 of 380 and 560 parts per million, so ambient and 1.5 times. So they're exposed, leaves are grown this way at 2 different CO2 concentrations. Then they're exposed to high temperatures, somewhere between 43 and 48, depending on the species. And as I said, this is a temperature where if you hold the leaf there, you see a steady, gradual decrease in photosynthesis. So photosynthesis becomes unstable at those temperatures that we're treating them at. So we treat them for only 15 minutes and then return them back down to normal temperatures. So we stress them either at 380 or 560, so we can separate the effects of growth and exposure responses to CO2 concentration. So the gas exchange measurements were made at 29 degrees before and after the stress, as well as during the stress period. So the first example of this is what we might think of as an expected result, and this is in soybean, glycine max. And here we get equal decreases in photosynthesis and stomatal conductance, equal relative decreases in photosynthesis and stomatal conductance, but a smaller effect at elevated CO2 during the stress period. So here we have photosynthesis treated over control and grown at ambient or elevated CO2. And we can see that if they're treated, which are the red bars, if their treatment CO2 condition is 560, their photosynthesis is less reduced than if the treatment condition is 380 parts per million. So high CO2, in this case, during the treatment period, is preventing some damage. And the same is true for stomatal conductance. And as I said initially, the relative changes in photosynthesis and stomatal conductance were essentially equivalent. And again, we see that when the plants were treated at ambient CO2, the inhibition of stomatal conductance is larger than when they're treated at 560. So this is sort of what we sort of long expected, and some of the other early literature suggested would happen. So that's the case of soybean. If we look at wheat, triticum, we get exactly the opposite response. And here we have larger decreases with elevated CO2 during the stress period. So here, during the stress period, again, we have photosynthesis treated over control, grown at either ambient or elevated CO2. But you see the red bars for either growth condition, where the exposure is at 560 parts per million, have lower photosynthesis, more reduction in photosynthesis than if they're treated at ambient CO2. So this is exactly opposite from what happened in soybean. And the same is true, the change in stomatal conductance. Here, stomatal conductance during this heat stress, wheat just opens up its stomates during heat stress. So these are higher than controls, but they're a little bit less here when the treatment is at higher CO2. So in terms of photosynthesis response, in wheat, we're getting larger decreases when elevated CO happens during the stress. Another species gives another totally different result, and this is for maize, Zea maize. Here, the elevated growth CO2 results in more damage. So you can see that it doesn't matter too much whether the treatment CO2 is 560 or 380, but the growth CO2, the elevated growth CO2 results in more decrease in photosynthesis during the high temperature stress. And in the case of maize, no effect at all on stomatal conductance at temperatures that decrease photosynthesis. So obviously it's a complicated response, and to make things even more complicated, we'll look at what happens in bean, Phaseolus vulgaris. Here we get effects both of growth CO2 and exposure CO2. Both of them have an impact. So if we're looking at photosynthesis, treated or controlled, for the ambient grown plants, then the high CO2 during the treatment causes more inhibition of photosynthesis. But if they're grown at elevated CO2, then the treatment at ambient CO2 causes more inhibition of photosynthesis. And the stomatal conductance responses are pretty much parallel to that. So 4 species for essentially different kinds of responses. So what kind of conclusions do you get from 4 species that are doing things totally different from each other? It's a little difficult. The C4 species, elevated growth CO2 tends to increase the damage. And this is what that early reference was saying that was true in the C4 species that they looked at. So growth at elevated CO2 in C4s, it looks like growth at elevated CO2 increases the damage to photosynthesis caused by high temperature. What I haven't talked about is what is the cause of that reduced photosynthesis. And from what I have shown you all previously, it's obviously not all stomatal conductance problem. There is some biochemistry, and there is actually some data from Co2 where he looked at several of the C4 photosynthetic enzymes that are specific to C4, like pepticarboxylase and some others, and they are actually inhibited more in plants that have been grown at high CO2, more inhibited by high temperature stress. C3s obviously just have a variable response. In some cases, high CO2 helps. In other cases, it hurts. In none of the cases is it really a stomatal conductance problem. And that's because in C3s, high temperature mostly eliminates CO2 effects on stomatal conductance. So that's not primarily what's going on here. It's photosynthetic problems, and they're just very variable as to how it's going to impact them. So just another hint that there's a lot of work that remains to be done. So I'm going to talk about chilling stress, CO2 effects on photosynthetic responses to chilling stress. And as I said, this is mostly motivated by the fact that in temperate climates, in order to avoid mid-season high temperatures during early reproductive phases where yield is very sensitive to high temperature events, farmers in temperate climates are tending to try and push earlier and earlier planting dates. And when you do that, one of the limitations you run into is chilling stress. So chilling stress, those of you from the tropics may not be very familiar with this. Species susceptible to damage at low but above freezing temperatures, generally sort of defined between 1 and 10 degrees Celsius. Often what happens is that chilling sort of locks the stomates open. So water deficits are often the cause of damage during chilling stress. And it's more common in species of tropical origin rather than temperate origin. But as crops, we tend to grow lots of tropical crops in temperate climates these days, like maize and soybean and phaseolus and things like that. So the hypothesis is that elevated CO2 partly closes the stomates and may result in less damage if the damage is caused by a water deficit. And this was sort of, this idea was that elevated CO2 may protect from chilling damage. The first time I saw it was a paper by Boza. I'm not sure how to pronounce his name, but this was in Plant Physiology. And he looked at bean and cucumber and corn, which is maize, and looked at photosynthesis at, it's hard to read, before, during, and during recovery are the three side-by-side comparisons. So the open bars are during the stress. The solid bars are recovery from stress. And the first three are for plants at 350 parts per million, and the second three are for plants at 700 parts per million. So you can see that in all three of these species, there's less damage. The open bar is higher here at the 700 than at the 350. And that's true in all three species. And it's also true in recovery for some of these species that after recovery is better when the treatment was given at 700 than at 350. So this was the basis for thinking that high CO2 may protect from chilling damage. And they thought that at least for bean and cucumber, but not in corn, that it was due to less water stress occurring when the CO2 was elevated during the chilling. And you can see that by what he's plotted here is relative water content for plants at 350 or 700 parts per million at different time before and then at different times during the stress and then after recovery. And you can see at the end, the 24 hours of chilling stress, there's lower or relative water content when the plants have been kept at 350 than when they were kept at 700, and in both of these species. Now, this did not occur in corn, but it did sort of support the idea that high CO2 may be protecting from chilling damage by keeping the leaves wetter. So one limitation to this is that the low temperature treatments in those cases were applied during light when the stomates were already open. And that's not the way chilling stress actually happens in the field. It happens at night in the field. Low temperatures, are pre-dawn. So in this experiment, what I decided to do is look at this, trying to apply the chilling treatment in a more natural way. So in this experiment, plants were grown at 2917 Celsius at 380 or 560 parts per million, given high light for 12 hours per day. And then the chilling treatment, again, we did both at the growth conditions or at the opposite CO2 conditions. So we can separate effects of growth CO2 versus CO2 during the treatment, during the chilling treatment. So what we did with temperature was we ramped during the dark period, we ramped down to 5 degrees Celsius, starting after the lights went out. Then it was held at 5 degrees for an hour in the dark and held at 5 degrees for an hour in the light, and then ramped back up to the normal daytime temperature over the course of about 3 hours. So this is a more natural, closer mimic of natural chilling event in this case. So we measured assimilation rate, stomatal conductance, leaf water potential in the light at 5 degrees. So while they're held at 5 degrees or for an hour in the light. And after 1 hour after returning the plants back up to 29 degrees. So we looked at photosynthesis, stomatal conductance, leaf water potential at the low temperature, and then after returning them to the light. So in soybean, we see that consistent with what Boza found, that stomatal conductance, look here at the right-hand side first, stomatal conductance during the treatment, if the treatment was applied using 560 parts per million, there was a larger reduction in stomatal conductance. Stomatal conductance was lower when the treatment was applied, when the chilling treatment was applied at 560, than when the chilling treatment was applied at 380. So not surprising. A high CO2 is closing the stomates partly, and it's making the leaves wetter. So leaf water potential is starting at 0 and going down. So higher water potential in plants that were actually dosed at 560 because the stomates were partly closed. So leaf water potential is not as reduced. So it is preventing some reduction in leaf water potential. So what happens to photosynthesis? So this is after chilling, and we're back up at our controlled daytime temperature of 29. And we see that even though the treatment at 560 parts per million kept the leaves wetter during the stress by having lower stomatal conductance, it did not prevent damage. There's actually more damage when they were treated at 560 parts per million when the chilling happened at high CO2 than when it happened at low CO2. And there's not much effect of growth CO2 on this. And the same is true for stomatal conductance after the chilling back at 29 degrees. It's much more reduced when the treatment happened at 560 parts per million than when it happened at 380. So in spite of the fact that high CO prevented the reduction in water potential, it actually still resulted in more damage in this case than at ambient CO2. So this is sort of counter to what we were expecting to find. So again, the story changes with species. So this is rice, or Oryza sativa. Again, after the chilling, back here measuring at 29 degrees. Here, the growth CO2 does protect photosynthesis. It's less reduced here than when the growth CO2 was ambient. And here, it's a growth CO2, not a treatment of CO2 effect, as opposed to soybeans, where it was the treatment CO2. So here, whether they were exposed to the chilling at 380 or 560 didn't make much difference. But if they were grown at ambient CO2, there was a larger reduction in photosynthesis by the chilling than if they had been grown at elevated CO2. And the same is true in stomatal conductance in this case. Larger reduction in stomatal conductance when the treatment was applied to plants grown at ambient CO2, regardless whether the treatment itself was at 380 or at 560. So we did also look at Phaseolus and Zea mays. And talking now about water potential, stomatal conductance during the chilling events. In rice, there was no CO2 effect on either leaf water potential or stomatal conductance during the chilling event, in spite of the fact that plants, in the previous slide, we saw that the reduction after the chilling event was greater in ambient plants. In Phaseolus, there's no growth or treatment CO2 effect on the inhibition of photosynthesis. Assimilation rate was reduced by about 33%, which was caused by a big reduction in stomatal conductance of 67%. So Phaseolus is quite sensitive to the chilling, but there was no effect of either growth or treatment CO2 on the inhibition. And in ZMAs, there again, no effect of either growth or treatment CO2 on the inhibition of photosynthesis. And in Maize, assimilation rate and stomatal conductance were both reduced only by 10 to 12%. So by this treatment at 5 degrees. So ZMAs is obviously much more tolerant of chilling conditions than any of the other 3 species we looked at. And probably not coincidentally, ZMAs is actually planted, even though it's tropical in origin, it's fairly cool tropical origin, and it is actually in this country, it's planted earlier in the spring than any of these other species, because it is fairly tolerant of chilling. So conclusions about the chilling stress is when chilling begins in the dark, elevated CO2 increase the leaf water potential in soybeans by closing stomates. But assimilation rate and stomatal conductance after recovery from chilling were still more reduced at elevated CO2. So elevated CO2 during the chilling actually resulted in more damage to photosynthesis and stomatal conductance after the chilling event was over. In rice, growth at elevated CO2 protected assimilation rate and stomatal conductance from the chilling damage, but it did not do it by improving leaf water potential, because that was not effective. And in maize and beans, there was no CO2 effect on chilling damage, which was fairly substantial in bean and very little damage in maize. And that's the end of my presentation.
Sorry. Thank you, Dr. Banks. Excellent presentation. We enjoy a lot. And very interesting, these aspects of photosynthesis regulation under high temperature and low temperature, and this involvement, this relationship with high CO2 concentrations. And this is come to the very center of our symposium, that are the climate change. And seeing your presentation, I was wondering, because always we talk about climate change, and always we think on increases in temperature and how this could affect plants, but you show here very clearly that this is not so simple, because in some C3 species may be this high temperature in combination of high CO2 concentrations can be even good for these plants, and sometimes can be very bad for C4 species. So think on it. I was wondering here, because there are several projects going on, and some of them very remarkable, related to transform C3 species, some C3 crops in C4. So in this perspective, and the possibility to transform the C3 crops on C4 maybe produce a bad aspect for these plants and create a new problem that you don't have. So my question, it would be more likely to hear your opinion about these possibilities, and I don't know, maybe transforming some species, some C3 species in C4 is too soon, in your opinion, or not?
That's a good question. One comment, that one thing that has always occurred to me about the effort to transform C3s into C4s is this is probably probably a pessimistic point of view, but is why don't we just wait for CO2 to go up? Isn't that what C4 is all about? is giving photosynthesis at high CO2. So in other words, C3s at high CO2 are better than C4s at high CO2. So why go? And if CO2 is going up anyway, why make the effort to convert C3s into C4s? Because at high CO2, they're worse off than C3s would be. So that's kind of a cynical point of view, but I do think that C4s have sometimes more problems at high temperatures than some C3s do, and whether that's inevitable in C4s, I don't know. I don't have enough experience with enough diversity of C4s. Certainly there are C4s that are very well adapted to hot conditions, and presumably they have ways of overcoming that problem. But things like Maize are not particularly adapted to hot conditions.
Yes, and maybe, Dr. Bounce, we can talk also to maybe, I don't know, I'm just talking, but oversimplification, because as you show here, when you interact two different environmental factors, you have completely different new scenarios, and sometimes good, sometimes bad. So in the end, what we have is a complex combination of different scenarios. So it's complicated to just predict what's the best for each, for everything.
Yes.
Okay. We have some questions from the audience. So asking you here, please. Can you hear Dr. Burns? But I can hear. Sorry, I can hear.
I see the question.
Okay. What's your personal idea, hypothesis by which some C3 plants seems to be more adapted to higher levels of CO2 in relation to C4 plants during stress exposure?
Yeah, that's a good question. I'm not sure how to answer that. In some ways, C3 plants started out evolutionarily in a very high CO2 world. And it's possible that still has some lingering effects on how they respond to it. Whereas C4 photosynthesis evolved when CO2 concentrations were quite low, and that was what spurred their evolution. Whereas C3s, evolutionarily, 1,000 parts per million is fine for them. So in that sense, maybe that has something to do with how they respond to what we're seeing now.
Okay. Indeed, there are three different questions from João Augusto. Thank you, João Augusto, for your question. So this is the second. Could you imagine the possibility of improving C4 plants using this mechanism, or even to use that improve the C3 plants that doesn't present this better adaptability?
Improving. Well, it's, at this point, it's a little bit unfortunate that the prime example of C4 photosynthesis that's been studied is Maize. And as I said, it's actually came from a fairly tropical but warm, not hot, mild environment. So it's not probably a very good example of a C4 adapted to a hot climate, whereas sorghum is probably a better reference point for that. But unfortunately, there's been very little work on photosynthetic responses to high temperature in sorghum that I'm aware of, anyway. And then, we have grain amaranths, which are C4s. And again, there's been very little work that I'm aware of on how they respond to high temperatures. So yeah, I think there are probably better examples of C4s being adapted to high temperatures than maize is, and I think maize is probably, unfortunately, that's the one that most of the work has been done on. I'm not sure whether that is.
Okay. And do we have one more from John Augusto? It's true, Nivea? Can you show us? Okay, so like in the case of soybeans and rice, that present such different patterns when exposed to killing stress. It's related to the previous questions, indeed, about the difference on C3 and C4.
Yeah, I have no explanation for why rice and soybean would respond so differently. They're not from such different places. I think we're going to find out when more studies like this are undertaken, that there's just a whole wide range of possibilities. And as was obvious from what I said, we're still a long way from doing any substantial biochemistry to figure out what on earth is going on. Gas exchange is one thing, but it's not a biochemical mechanism directly. So I think there's just a whole lot to learn. I'm sort of hoping I'm pointing people towards things that are interesting to explore, not that I have any explanations for what's going on.
Thank you, João Augusto, for the interesting topic. We have another question, Nivea. Okay, it's from Ana Carla, one of our organizing committee members. Great talk. What is known about the leaf temperature in these plants exposed to this stress?
Well, in the high temperature stress part of this talk, those were the exposures were based on measured leaf temperatures, not air temperatures. So those are specific to leaf temperatures. And in the chilling stress, obviously the chilling stress was the 5 degrees was imposed in darkness. So those leaf temperatures should be essentially air temperatures in darkness. They warm up a little bit when the light comes on, but probably not very much. And those are very well ventilated growth chambers, so it's probably not much above. But certainly in the dark, they're going to be right at the air temperature. So I don't think that's a major issue with what I presented. Certainly, when you're talking about field work where you're trying to expose plants to, say, high temperature treatments combined with elevated CO2, their leaf and air temperatures in the field can be quite different from each other, and there you have to decide what your treatment really wants to be. And the ones that I'm familiar with, where they've combined high temperature treatments with elevated CO2 in free air systems with the overhead infrared heaters, there they base it on controlling leaf temperature, but they don't control leaf temperature the same at an elevated and ambient CO2 plot. So in other words, the elevated high temperature treatment is a temperature that's based on measured temperatures at elevated CO2 without any heat treatment. So those run a little bit warmer than ambient CO2 leaves. So when you compare high temperature treatments between ambient and elevated, you're not actually comparing the same leaf temperature treatment. So it's something to think about. In our experiments, those were based on leaf temperatures, but that's not always the way it has been done. It's not always the way it necessarily should be done either.
Yes, in fact, Ana Carla has completed the question here. I mean, because some species decreased GS at high CO2, and I think this increased leaf temperature negative effect. Yeah.
Yes.
Yes.
Yeah. What I found in the C3s that I looked at when they were exposed to high temperature, then temperatures at that range, I did not find differences between. I didn't find CO2 effects on stomatal conductance. That difference that you normally see tends to disappear when you get up to temperatures that are high enough to stress photosynthesis. So that's for. I would say that in that case, the high temperature treatment is essentially wiped out CO2 effects on stomatal conductance in these experiments.
Perfect. Thank you, Ana Carla, and thank you, Dr. Burns, for your response. Dr. Burns, I was wondering also about these experiments of high temperature and high CO2. And we see a lot of experiments that are performed in growth chambers on a constant temperature or a constant increased CO2 condition. And thinking on the first question I made, that the complexity of the environment is not always the same, and this complexity and the interactions between the environmental conditions and the plant response can produce new scenarios, unpredictable. So I would like to hear about you, what you think about this comparison between these results of constant increased temperature with some other kinds of approach with oscillating temperature experiments and this kind of approaches, and also with CO2, with phase experiments compared to growth chamber experiments. I would like to hear your opinion about that.
Yeah, difficult questions. In terms of CO2, I think, current ambient CO2, at least in our environment, changes substantially day versus night. What you call ambient CO2 could be 500 parts per million at night and closer to 400 during the day. But the nighttime is very variable depending on the wind speed. But daytime CO2 concentrations out in the open field don't fluctuate very much. They may drift by 10 or 20 parts per million during the course of a day, but they're not wildly changing. But when you try to apply elevated CO2, it's very difficult to get that smooth a control of CO2. So elevated CO2 treatments normally are quite variable. And I do think that has an impact on the plants. And temperature, I think you have a better chance of controlling temperature than you do CO2 in terms of smoothing things out. But then the question is the target. It's obviously much more realistic to have sort of normal diurnal patterns of temperature rather than a constant fixed temperature during the day. And that's that's fine. I think it depends on the specific objective of the experiment, whether you can deal with that complexity or whether you can't, and you just need to eliminate it. If you go with varying temperatures, then you have all the questions of the magnitude and the rate and all that, and how do you pick a standard to compare with? So, I think you have to look at the specific objective of each experiment to decide whether constant or varying for either CO2 or temperature.
Yes, I agree totally, and especially when I think to bring this information to the field, I think that this is a big, very important thing to worry about. We have one question from Ana Carla, another question from Ana Carla Nivea. Thank you, Dr. Burns. I'm interested to know what has been PP system developing to help us better understand these gas exchange response in plants. Oh.
Well, PP systems, let's just say portable photosynthesis systems in general have, I think, made a huge step in being able to measure photosynthesis under realistic field conditions. And that has the ability to do that, and then to be able to control specific conditions has just improved over the years as these instruments develop more capabilities. So one of the newer developments of PP systems is the ability to do these rapid ACI curves, which Lycor has made a big deal about doing, but you can also do that with the Cyrus-3 in PP systems. You can actually do it faster than Lycor can do it at this point, because we have at the talk by Lycor, they were still recommending that the rate of increase in CO2 is no more than 100 parts per million per minute, whereas we can double that rate, so we can get curves done in half the time that they can with a PP system. But response times of the instrumentation and PP system has, rather than go with the open path CO2 analyzer, still has closed sealed cells and brings air samples back from a leaf cuvette to sealed cells. But they still have faster response times than Lycor, just because the volumes of the cells and the sample chamber are much smaller. So the response time depends both on a lot on the volume of the whole system. So that's been PP Systems' direction, is to get fast response times by making the leaf chamber and the CO2 analysis cells small volume. So there are other, always looking at ways to improve things. But it's kind of funny. We still get inquiries of people who, for what they want to do, would be better off with the old handmade thrown together systems, because they want to deal with large leaves and large volumes and large flow rates. Whereas the point of the portable systems is to be small. But there are some very legitimate research purposes where that is not what you really want. You want a big, slow responding system where you can expose a large amount of leaf material. So needs keep changing. Instrumentation keeps improving.
So, thank you very much Thank you very much, Dr. Burns, for explanation. Thank you for the audience for very nice questions and a very interesting discussion we had today. Um, I would like to now to make some comments about our scheduling. We have in a few hours the second session of oral presentations from works that people sent to us, and expect to see all of you there. And we have two sessions indeed today. So this is our last presentation from the special invited speakers. Now we have excellent speakers, but they are related to works that are sent to our symposium. That are excellent works, all of them. So I would expect to see a lot of you. And I would like to say thank you, Dr. Burns, once more. And thank you to PP Systems and Lab Control for this possibility. And please, Dr. Burns, if you have some final remarks to say for us.
Well, I congratulate you on a very nice symposium. It's been, I've managed to see several of the talks, and everything has been very, very well done, very well organized. Thank you.
Thank you very much. So, bye, guys. See you soon.